Poison dart frogs exhibit vibrant colors as a warning signal to potential predators. This phenomenon, known as aposematism, deters predators through visual signaling. The bright coloration indicates the presence of potent toxins within the frog's body. Aposematism is an evolutionary adaptation that enhances survival rates. Predators learn to associate the frog's colors with the unpleasant or harmful effects of the toxins. The effectiveness of aposematism depends on the conspicuousness of the warning signal. Poison dart frogs secrete batrachotoxin, a highly potent chemical defense mechanism. Batrachotoxin's high toxicity can induce paralysis or cardiac arrest in predators, even in minuscule amounts. The chemical structure of batrachotoxin features a complex arrangement of rings and functional groups. Batrachotoxin functions by disrupting sodium ion channels in nerve and muscle cells. The toxin's mechanism of action leads to a cascade of physiological effects, culminating in paralysis or cardiac arrest. Minute quantities of batrachotoxin are sufficient to cause significant physiological damage. A single golden poison frog contains approximately 2 milligrams of batrachotoxin. Batrachotoxin is an extremely potent neurotoxic steroidal alkaloid. The concentration of batrachotoxin within the frog's body is remarkably high. 2 milligrams of batrachotoxin is a lethal dose for most small animals. The golden poison frog uses batrachotoxin as a defense mechanism against predators. Batrachotoxin functions by irreversibly increasing the permeability of the sodium channel of nerve and muscle cells. Certain indigenous tribes historically extracted poison from dart frogs. The extracted poison was applied to the tips of blowgun darts for hunting purposes. Poison-tipped darts created lethal weapons for hunting. This hunting method provided a significant advantage in acquiring food resources. The use of dart frog poison represents an ingenious application of natural resources. Poison dart frogs raised in captivity are generally non-toxic. The absence of toxicity indicates that the source of the poison is not inherent to the frog. The toxicity of wild poison dart frogs is derived from their diet. Dietary alkaloids are sequestered by the frog, resulting in the production of skin toxins. Captive diets lack the specific insects needed for toxin production, hence the non-toxic nature. Poison dart frogs obtain toxins from their diet in the wild. The toxins are derived from specific alkaloids present in ants, beetles, and mites. These alkaloids are sequestered by the frog and stored in its skin glands. Dietary sources of alkaloids are essential for the frog's toxicity. Without these dietary alkaloids, the poison dart frog would not be poisonous. The frog's body converts alkaloids from prey into potent toxins. This conversion process represents a unique chemical adaptation within the frog's system. The stored toxins serve as a defense mechanism against predators. Alkaloid conversion and toxin storage showcase a complex biochemical pathway. Poison dart frog species exhibit varying levels of toxicity. Toxicity levels range from mild skin irritants to deadly toxins. The golden poison frog contains some of the most potent toxins known in the animal kingdom. Different toxins affect the nervous system in varying degrees, leading to paralysis or death. Toxicity differences are attributed to variations in diet and toxin accumulation mechanisms. The golden poison frog of Colombia is considered the most poisonous animal on Earth. Extreme caution is required when handling golden poison frogs due to the risk of accidental exposure. The frog's toxicity poses a significant threat upon contact. The poison is a complex mixture of toxins that can cause paralysis and death. The frog's bright coloration serves as a warning signal to potential predators. The poison is used by indigenous tribes for hunting. Frog toxin, when replicated, exhibits immense potency. A teaspoon of replicated frog toxin could potentially be lethal to over 200 people. The high lethality underscores the significant danger posed by such toxins. Replication processes can exponentially increase the volume and impact of natural toxins. The potency of replicated toxins necessitates stringent safety protocols and research oversight. Deforestation destroys the natural habitat of poison dart frogs. Habitat loss reduces poison dart frogs' access to essential food sources. The destruction of the rainforest ecosystem directly threatens the survival of poison dart frogs. Fragmentation of habitat isolates frog populations, limiting genetic diversity and increasing vulnerability. Climate change impacts the rainforest ecosystem. Changes in temperature and rainfall can disrupt the rainforest's food chain. Climate change may alter the availability of toxin-producing insects, a food source for frogs. Disruptions in the food chain and toxin availability can negatively affect frog populations. The illegal pet trade decimates poison dart frog populations by extracting them from their native habitats. Removing poison dart frogs from their natural environment further endangers their survival. The illegal pet trade directly threatens the biodiversity and ecological balance of rainforest ecosystems. Unregulated collection practices often lead to high mortality rates during transportation and captivity. The demand for exotic pets fuels the illegal trade, jeopardizing the long-term survival of poison dart frog species. Scientists are exploring frog toxins for potential development into novel painkillers. Frog toxins possess unique chemical properties that may be harnessed to alleviate pain. The research aims to leverage these natural substances to create more effective pain relief medications. The unique properties of frog toxins offer a promising avenue for innovative pain management solutions. Research explores the potential of frog toxins for treating heart conditions. The aim is to harness the effects of frog toxins on the cardiovascular system. Frog toxins may offer novel therapeutic pathways for heart disease treatment. Specific compounds are isolated and analyzed for their impact on cardiac function. Frog toxins are being studied for their potential in creating antidotes to other poisons. The complex chemical structures of frog toxins enable them to counteract harmful substances. Scientists are developing antidotes by leveraging the unique properties of frog toxins. The research aims to understand and replicate the mechanisms by which frog toxins neutralize poisons. Antidotes derived from frog toxins offer potential treatments for various types of poisoning. Captive frogs can lose toxicity compared to their wild counterparts. The loss of toxicity in captive frogs raises concerns about the survival of wild populations. Environmental changes can affect the survival of wild frog populations. Reduced toxicity may impact a frog's ability to defend against predators. Changes in toxicity levels can disrupt the ecological balance. Environmental changes can trigger the evolution of novel toxins in animal species. The emergence of new toxins can alter the established dynamics within a rainforest ecosystem. Evolutionary adaptations to environmental stressors may lead to the development of defense mechanisms, including toxins. Changes in toxin profiles can have cascading effects on predator-prey interactions and overall biodiversity. The introduction of new toxins can disrupt the balance of the rainforest ecosystem. Scientists are investigating poison dart frog alkaloids for potential pharmaceutical uses. Alkaloids from poison dart frogs possess unique chemical structures that may interact with human physiology. Research focuses on synthesizing analogs of these alkaloids to enhance therapeutic effects and reduce toxicity. These alkaloids can potentially be developed into novel drugs for pain management, neurological disorders, and other conditions. The extraction and purification of alkaloids require advanced techniques, such as chromatography and mass spectrometry. Ethical considerations are paramount when studying and utilizing compounds derived from endangered species. Conservation efforts are essential for protecting the habitat of poison dart frogs. Habitat preservation ensures the survival of these unique and valuable species. Maintaining biodiversity is crucial for the health and resilience of the rainforest ecosystem. Protecting poison dart frogs contributes to the overall ecological balance of the rainforest. Poison dart frogs synthesize alkaloids into toxins through a complex biochemical pathway. The exact mechanisms of alkaloid synthesis in poison dart frogs are not yet fully understood by scientists. Researchers are actively investigating the specific enzymes and precursors involved in the alkaloid synthesis process. The biochemical pathway involves multiple enzymatic reactions and molecular transformations. Understanding this process could provide insights into pharmacology and toxin production. Further research is needed to fully elucidate the complete biochemical pathway of alkaloid synthesis. Despite their toxicity, poison dart frogs are still vulnerable to certain predators. Some predators have evolved resistance to the toxins of poison dart frogs. Evolutionary adaptation allows certain species to consume poison dart frogs without harm. Predator resistance mechanisms can involve specialized proteins that bind to and neutralize the toxins. Genetic mutations can lead to physiological adaptations that confer toxin resistance. The coevolutionary arms race between predator and prey drives the development of increasingly potent toxins and resistance. Genetic studies aim to identify genes responsible for the frog's toxin storage and conversion capabilities. Researchers are analyzing the frog's genome to pinpoint specific genetic sequences related to toxin processing. The process involves mapping the frog's genetic structure and comparing it to other species. Gene editing techniques may be employed to validate the function of identified genes. Understanding these genes could lead to advancements in detoxification methods and drug development. The research seeks to uncover the genetic mechanisms that enable the frog's unique adaptation. The frog's diet consists of various insects, including ants, beetles, and mites. Key alkaloids are present within the ants, beetles, and mites that constitute the frog's food source. Close examination reveals the specific types and concentrations of alkaloids found in these insects. These alkaloids play a crucial role in the frog's physiological processes. The presence of alkaloids in the frog's diet directly influences its survival and adaptation. Batrachotoxin features a complex steroidal structure with multiple fused rings and functional groups. The unique arrangement of atoms in batrachotoxin contributes to its potent neurotoxic effects. Alkaloids found in poison dart frogs exhibit diverse chemical structures beyond batrachotoxin. These alkaloids share common structural motifs, such as nitrogen-containing rings, which influence their biological activity. The spatial arrangement of atoms in these molecules is crucial for their interaction with biological targets. Visualizing these chemical structures aids in understanding their synthesis, properties, and mechanisms of action. If you enjoyed this journey through Mind Jungle, make sure to subscribe and join us for more mysteries of nature, history, and the world around us.